being podcast, which is very unnerving because it means that there are things I really oughtn't to say because they might go out on the web. And I do wish that my presentation this afternoon was as well organized as allegedly those finals papers were back in 1972. I fear they may not be. There are lots of, I can't say old friends anymore, friends of long standing in the audience, and I'm delighted to see them. And may I particularly acknowledge Professor Charles Holland and George Sevastopoulos. Those two professors had much to do with where I ended up going. So they can take the blame or the credit as you prefer. So it's very nice to have them here this afternoon. So I'm at the Peabody Museum in New Haven, and those of you who haven't had the privilege of visiting, I invite you. It's a great place to be. We have a fabulous full-size statue of a Peabody dinosaur, Torosaurus, outside the museum. This is it here on Whitney Avenue, which in the last 5 years since it was erected has become a kind of New Haven, Yale landmark or icon. Inside the museum we have this famous Zallinger Age of Reptiles mural, which will be familiar to some of the older people in the audience because, of course, it was figured, and indeed is still figured, in lots of popular books about dinosaurs. It's now way out of date, but a very interesting teaching tool in terms of using it to illustrate how science has advanced since. And we have a great hall of dinosaurs that also dates from the 1940s, and if anybody happens to have $10 million in their back pocket, I'm trying to raise money to redo it at the moment. I hope that's on the podcast. We have about 12 million specimens. We always say provisionally, and I say provisionally because, of course, the argument is that I'm still counting them. And they run in 11 different divisions. So the Peabody runs from anthropology right through to meteorites and indeed historical scientific instruments. And although our displays, at least the dinosaur hall itself, is somewhat dated, we essentially have state-of-the-art collection facilities for the most part. So it's an interesting place to come and visit and do some science when any of you would like to do that. So I'm going to talk about exceptionally preserved fossils. And of course, one of the matters that inevitably exercises us when we talk about fossilization is decay. So this is this morning's decay slide. As you can see, it says the maestro is decomposing. And as he's presumably received an honorary degree, otherwise he wouldn't be on the screen today, I've given him a D Lit because he's clearly a musician. But you'll notice that he's losing some of the soft tissue on his arms, and he's clearly been doing this for some time. So we can look at decay, and I'm going to give you a very brief 101 on decay, so bear with me, those of you who are very familiar with this, just for a few minutes so that you have some background. We can look at decay in the fossil record itself. So a series of fish fossils, this thing Priscacara, comes from the Eocene Green River Formation in Wyoming. I'm sure I'm driving these guys nuts because I keep moving around. So you can watch it decay with a series of specimens on the lake bed until we get to examples like this, which have quite clearly exploded due to the buildup of decay gases on the floor of the lake and scattered the various pieces around. So quite clearly, that shift from soft tissue outline to skeleton implies decay prior to fossilization. How do soft tissues survive? How do these non-biomineralized parts survive? Well, there are two essential processes. One of them is organic decay. And I can illustrate that with a series of slides based on a famous deposit called Clarkia in the late Miocene of Idaho in the northwestern United States. And here we have a classic example of organic preservation. When you work through this material, you find leaves on the surfaces of these slabs. You can take these leaves into the lab, and this is something that comes with a health warning. Don't try this at home. This is Bill Rember, who's a paleobotanist of long standing and very cavalier with hydrofluoric acid. Not advised. But you'll see he sticks his... It's not quite what it seems. He's put acid on the surface of the slab, and then he's now using a pointer to tease off the leaf from the surface. And then he mounts them between two sheets of acetate, and you can hold them up to the window. Here's a rather nice example. So obviously this is carbon, essentially carbon compounds, which have survived, in this case, to the Miocene. Of course, there are lots of examples that go back much, much older. Were it not for this process, the paleobotanists would be largely out of work. So organic materials survive because they're selectively preserved, they're resistant to decay, and they also modify diagenetically through the rock record in a way that I'm not going to talk about today because it would take too long. So that's one half of the story. So we can look at decay in different fossils. We can also look at decay in the lab. And these are the kinds of experiments one needs to do before lunch rather than after lunch. So this is a series of worm slides. It's just a worm in a petri dish, a polychaete worm, a bristle worm. And you can actually, as many of us have shown, get quite elegant results out of this kind of observation. So in 2 days, the back of this thing has gone essentially to fluid. It begins to leak. Within 6 days, it's largely flattened. And then we end up very shortly with some kind of remnant of the outer cuticle. Of course, the bristles survive. And if you run this thing for as long as any research grant will allow you, you'll end up with many of the bristles that give the bristle worms their name, but also the jaws. These are organic. This is decay-resistant organic material, which also has a very high fossilization potential. But what of the rest? What of all those smelly soft tissues? Well, if those are going to survive, they have to be mineralized very rapidly at a very early stage in the process. And one of the classic examples of this kind of mineralization is based on the Cretaceous Santana Formation of Brazil. The work was done many years ago now, initially by David Martill, who's at Portsmouth. This is clearly an odd fossil fish, not because the fins have got knocked off, but because it's three-dimensional. Because, of course, as the worm experiments show, soft tissues decay and soft-bodied organisms collapse very rapidly. So normally fishes, of course, you'd expect to flatten out very quickly through decay. This is a three-dimensional fish, and it's three-dimensional because within these concretions, which retain and help to sustain the three-dimensional nature of these things, we get the remains of muscle tissue. And this muscle tissue is such that you can see the original fibers between the ribs of the fish, and this material is preserved in calcium phosphate in a mineral called apatite. And clearly, given what we know from our very simple worm experiment, this kind of mineralization has happened very rapidly indeed, initiated within a matter of days and weeks in order to retain these kinds of soft tissue information. So two things going on: decay resistance, and therefore carbon compounds on the one hand, and then the most labile, the most decay-prone tissues like muscles and guts, generally speaking, have to have experienced some kind of very early mineralization in the sediment. So that's what we're essentially talking about in terms of process. We can replicate this early mineralization in the lab, and these are slides showing a comparison with, the tissues that surround the muscle in a fossil fish. This is Rhacolepus, again from that Santana Formation in Brazil. Here are the sarcolemma, the sheaths that surround the fibers, and here is the banding on the fibers. These are, of course, scanning electron microscope pictures. And we can do this very same thing with shrimps in the lab over a period of just a few months and show that with the right kind of circumstances, this mineralization occurs very rapidly. So soft tissue fossilization, in a kind of counterintuitive way, involves very early processes that then set the stage for longer-term preservation. Okay, so what do extraordinary fossils inform us about? They do a number of things, and I'm going to try and illustrate these with a kind of series of case histories that I hope you'll find interesting. So firstly, they provide information on animals normally known only from their fossilized hard parts. So Ian mentioned finals. I thought it might be interesting to look at my finals paper. This is back in 1970. I don't know whether this is the one maybe I smoked cigars before all of them. I simply can't recall. This would have been the one probably George and Professor Holland set. So it's the moderatorship in Trinity term 1972 in paleontology. And actually we can see which 4 questions I did. But what I didn't do, interestingly enough, is number 2. Comment on the evolution of the conodont-bearing animal. And interestingly enough, not that long after, about 10 years after I left Trinity, I managed to resolve, with colleagues of course, like all this stuff, something about the nature of the conodont-bearing animal. And for those of you who aren't familiar with this story, conodonts are small microfossils, phosphatic elements, little teeth or tooth-like structures that are pretty much ubiquitous right through Paleozoic rocks and much beloved by biostratigraphers, people who correlate and date sedimentary sequences, because you can dissolve them out of limestones and you can extract them from shales. And for something like 125 or more years, everybody speculated endlessly on what these things belong to, literally from plant parts right through to all sorts of vertebrates with pretty much everything in between, mollusks and a whole variety of invertebrate taxa. The problem was the soft tissues never survived. These are animals that feed up in the water column for the most part, decay and sink to the bottom, and rarely survive. Back in the 1930s, people had discovered what we call bedding plane assemblages, associations of this type of element in an apparatus on the bedding plane, and that gave us some kind of clue. But in the early 1980s, I was fortunate enough to be involved with Ewan Clarkson at Edinburgh and Dick Aldridge, now at the University of Leicester, with discovering the first examples of the soft tissues of this animal. So here's a classic case history where we knew the hard parts, but we had no idea what the organism looked like. And ironically, this worm-like swimming or eel-like creature turns out, we're almost certain now, to be some kind of early vertebrate. So we could certainly have written a much better answer to that question now than we were able to in 1972, because the soft tissue story has filled in many of the gaps, and the conodonts now, depending on who you believe, sit down here somewhere basal of all the more advanced vertebrates. The jawed vertebrates would be up here. So the conodonts are down, with the hagfishes and lampreys in the vertebrate stem. So soft tissues filled out the picture. We could now say something about that conodont-bearing animal. And more recently, I've also been involved in solving another mystery, a much less interesting one perhaps, but shelly or platy fossils known as macaridians are found throughout, again, much of the Paleozoic, particularly in limestones, and normally as isolated plates like these brown structures here on the reconstruction. Again, nobody had any idea what they were. Barnacles perhaps, echinoderms, worms of various kinds. And we started recently working in Morocco with Paddy Orr, who's here from UCD, and a postdoc who was with him and is now with me, Peter Van Rooij. And with another graduate student, Jacob Winter, we were able to show that these extraordinary plates line up in a kind of arrangement like this, which was known, but we have the soft tissues. So this is actually some kind of polychaete worm with a series of scales, and it's essentially a bristle worm with a scaly back. So again, the soft tissues resolved a question that nobody was able to deal with simply based on the hard parts alone. So we have other examples of this kind of thing. Ireland has its own exceptional preservations, and they also fill out some of this story. I got interested in Limnostheria way back, probably as a postgraduate, because I was aware that it was here in Ireland. It comes from boreholes in the Kilkenny coal measures. And Paddy Orr and I wrote an extensive paper on it some years ago now. You can see that I bought this reprint from the Academy in 1976, intrigued by the appendages that Mabel Wright illustrated in this remarkable little monograph. And we tracked down the specimens. This is what a living concostragon looks like. This is obviously a female because it's got a lot of eggs within the carapace. We tracked down the specimens, and these are photographs that Paddy Orr took. And you can see the antennae here very nicely preserved in two specimens, including traces here of the claspers. This is a male concostragon, and you have to bear in mind that this is one of the best examples of these little bivalve crustaceans from the fossil record where we actually have some soft tissues preserved. And remarkably, they've been picked up just in the core of a borehole by splitting, and these materials were partly in the survey here, are partly in the survey here, and we're also in the Natural History Museum in London. So Paddy and I were able to figure out the head appendages of this thing, and indeed the tail. Here's the back end. You can see a little telson with a couple of limbs projecting out, and here's a modern equivalent just for comparison. And Paddy has done some superb work using elemental mapping on the microprobe to try and work out the composition of these things, the combination of perhaps clays and pyrite that are associated with the limb preservation, so that we can say something about the conditions under which these animals were preserved. Not something I'm going to talk about this morning. And there are other things from these boreholes. This is another very obviously little shrimp, in this case some kind of a syncarid, showing a beautiful tail fan. So there's a fauna dominated by concostrogans, but also with one or two other little arthropods as well. So Ireland has its exceptional preservations. So these are examples where we knew the fossils, we had hard parts, but we couldn't flesh out the organism because we only had a partial indication of what it was like. So what about revealing otherwise unknown animals? You'll appreciate that something like 60% of all modern organisms in terms of the variety of types are actually totally soft-bodied, so have virtually no good fossil record. So we're relying on these exceptional preservations to reveal those kinds of animals, and of course some of them are animals that have long gone extinct. So if we go back to the Cambrian, this is me in one of my favorite localities up in the Canadian Rockies at the Cambrian Burgess Shale. And here's a reconstruction of that community with all the interesting kinds of Cambrian creatures which are familiar from the literature, but not, of course, familiar by comparison with modern examples, including this giant predator, Anomalocaris, which is still exercising us in various ways in terms of new discoveries, which, well, watch this space. If you take away all the soft-bodied organisms in this kind of Cambrian reconstruction, this is your normal fossil record. So, as it says down here, about 86% of genera are gone, and potentially up to 98% of the individual animals in this marine deposit would have been or were soft-bodied. So this, if you like, is your normal fossil record. If you want a better illustration of why you should look at soft-bodied fossils, I can't really think of one. And of course, this was discovered back at the beginning of the last century by Charles Walcott, and it's revealed all sorts of extraordinary animals. And just by way of illustrating one of the unknowns, Opabinia is perhaps the classic. Walcott describes it here as some kind of worm-like organism. And this is my late lamented PhD advisor, Harry Whittington, who was still publishing until last year, I guess, well into his 90s. Paleontology is good for you, folks. This is his reconstruction of Opabinia. When he first showed it at one of these Paleontological Association meetings back in the 70s, I guess it was, the whole audience started laughing. Something that's totally inconceivable now because we've got so used to thinking about Cambrian animals as being kind of weird. But if anything, what more research is showing is that they're actually not that weird. They're certainly unexpected. Here's a rendering of Harry's reconstruction in Steve Gould's book, Wonderful Life. And here are some modern reconstructions. The color scheme here is courtesy of the Royal Ontario Museum. It has no basis in fact. We'll talk about color in fossils momentarily, but this is not a good example. But here are a couple of reconstructions of this animal. This is Graham Budd's with a series of little lobe-like limbs concealed beneath the flaps. And here's a more recent one by me and a Chinese colleague that rejects Budd's interpretation. And I show this simply to illustrate that Opabinia remains controversial. And here's a recent reconstruction from a website which is currently being put together by the Royal Ontario Museum by Jean-Bernard Caron. And you'll notice that the controversy regarding the lobe limbs that are hidden by these flaps is concealed by the nature of the reconstruction. You just, if you can't put the whole thing together, you hide the bits that you don't know about. And this is essentially what's going on here. So this is a classic unknown animal. But now, the point about this now is that with more modern phylogenetic methods, we can show that although it's weird, it's not so extraordinary because it fits in here very nicely just below all the major arthropods that we're familiar with today, the major groups like crustaceans and chelicerates and insects and so on. will fit here, and Opabinia slides just below that, separated from the so-called euarthropods, the true arthropods, by anomalocaridids, these big predators. So it's weird on the one hand, but now we've made progress and we know how it fits into the scheme of things in the way that we didn't before. So they fill out the morphology when you just know the hard parts. They provide information on animals like Opabinia. Without exceptional preservations, we just have no idea that Opabinia ever existed. It would be much poorer. Soft tissue preservations also provide a much more complete picture of ancient ecology. So back to my finals paper. So this one I did do, discuss the functional morphology of trilobites, probably set by Professor Charles Holland. We have recently, indeed with a TCD student, Una Farrell, who came from, she was my first Yale student, came from the geology department here, has now submitted and is waiting to defend. She worked on Beecher's trilobite bed, which of course is a classic locality originally discovered in the 1890s when gentlemen knew how to dress in the field, even in North America. So this is Beecher and cronies at his locality. These are why we're excited about Beecher's trilobite bed. Very spectacular trilobite material preserved in pyrite with not just the exoskeleton, but also the appendages. So Una has been working here. This is the classic Beecher restoration. And I guess when we were learning about the functional morphology of trilobites back in the 70s, this was one of the reconstructions of reference, or at least versions of it. Since then, Beecher's bed has had many revisits, and Triarthros in particular. Here's a reconstruction, or rather an X-ray by John Sisney, and here's a new reconstruction by Harry Whittington, my former advisor, and a student of his, John Ammond, published in the 1980s. But Una and I weren't interested in the trilobite's morphology per se, but actually in its life environment, where it was laid down and fossilized and what that tells us about its living conditions. So we went back, a couple of amateurs, we shouldn't call them amateurs because actually one of them was a PhD in chemistry who worked for Kodak up near Rochester in New York State, rediscovered the locality in the 1980s, and I went out there and did a kind of rescue excavation and did some geochemistry with a group at Leeds. And then it was all forgotten again until Una came along, and I thought, well, we should re-excavate this and really make some sense of it. So these are the landowners, Stan and John Koziarts. Stan is legally blind. He's not allowed to drive vehicles on the road, but he does operate these machines. So we try and keep at a very safe distance as far as possible when we're in the field with him. So this is our excavation. It runs for something like 80 yards along the riverbank. And this sort of Ordovician fine-grained sediment degrades very quickly, so the cliffs can tend to disappear and erode in. Now, what Una and I were interested in was looking at the conditions under which the trilobites were living and where the pyritization was taking place. So I'm not going to go into detail. These are distal turbidites for the sedimentologists among you, and we get trilobites at several levels in the quarry. These would be centimeters. And we can look in detail at slabs and indeed X-rays, and these are polished sections, although they're not very clear in this kind of light. So you can get a lot out of mud rocks, although they're tough to work with. This was a very challenging project. And she did a huge amount of geochemistry as well. The interesting story for us here today is that these kinds of trilobite beds occur at several levels. We were originally focusing just on the level that Beecher had discovered. And not only are they at several levels in the quarry where we've done our excavation and now collected in huge detail, they actually occur at many places in this formation, this Frankfurt Shale, throughout upper New York State. So these are a number of the localities. The original one, Beecher's trilobite bed, is here near a place called Rome, ironically. Here's Joe's quarry, which was discovered in 2008, which is not very far from the original Beecher's bed, but the Martin quarry was discovered by a guy called Marcus Martin, who in his real life I think works as an accountant or a solicitor and does this thing in his part-time, and has opened up a major excavation and is now still excavating at Beecher's trilobite bed, as it were, for us, and giving the museum the best specimens. These are the kinds of things that he's finding, really spectacular specimens If I stood in front of the light, you'd be able to see them better. But anyway, you get the idea. So the message here is that when you start doing collecting, you find that these kinds of exceptional preservations are really not rare at all. I mean, we always imagined that somehow Beecher's bed was unique. And now we discover it occurs at many levels at the original locality and at several levels up to 60 or 70 kilometers away in upper New York State. And of course, in this new locality, the so-called Martin Quarry, it occurs with a variety of other organisms which we don't see at the original Beecher's bed locality. So we can say more about the nature of trilobites. This little story is not from work that we've done at Yale. It's Jason Dunlop and other colleagues looking at the Riney Chert, which is a famous Lower Devonian locality at Scotland. But I throw it in as information on what we can do about ecology with exceptional preservation. The Riney Chert is really famous for plants, but it also yields the earliest insects or material of the head of an early insect, different kinds of insect groups. We won't go into those now. And these wonderful little spider-like animals called trigonotarbid. Here's the jaw of a trigonotarbid. And these things, this is a kind of hot pool where you've got siliceous material, a bit like Yellowstone or something like that, emanating into a swampy environment and trapping and preserving some of these remarkable little animals and the plants that they were living around. So here's what a trigonotarbid looks like. Looks for all the world like a spider, although it's related. It's not exactly a spider. But in these sections in the chert, which are translucent, you can see through them, you get extraordinary details of the respiratory structure. So this is from the work by Dunlop et al., and these are sections at a micron distance done by focusing a microscope down through this translucent chert, and then they've packaged that together to produce this kind of reconstruction. So this is the lung tissue from a Devonian spider-like animal, showing the internal features, including the pillars that separate the lamellae, and demonstrating absolutely definitively that this thing was air-breathing, which was always a slight controversy because, of course, it's critical. These are some of the earliest examples of terrestrial arthropods. So again, exceptional preservation, important ecological information. Let's talk a little bit about feathers. Archaeopteryx, famous as the earliest bird, let's put bird in inverted commas, but I'd be quite happy calling it a bird, from the famous Solnhofen limestone, Jurassic of Bavaria, first published in the 1860s. This is the London specimen of Archaeopteryx, and this is the one isolated feather that was discovered at about the same time. I'd have had trouble getting this into the story. The best I could do, perhaps, would be where I'll outline the main features of Paleozoic vertebrate evolution. Paleozoic vertebrate evolution, ironically now, would involve the evolution of the conodont-bearing animal because they're Paleozoic vertebrates. So questions 2 and 6 could have been answered with the same answer. George wouldn't have let me get away with that, but potentially it could have been done. So supposing we ignore Paleozoic, we can talk a little bit about Mesozoic and later vertebrates when we talk about the feather story. Well, the feather story started with one of my still current graduate students, Jacob Winter, who's a Dane. And Jacob was doing a class project on squids, fossil squids. And we've known for a long time that squid ink sacs preserve very well. They're nice big three-dimensional structures in many of these fossils. And we know that they preserve the melanin, or at least the morphology of melanin, because people had demonstrated that looking at them under the scanning electron microscope. So Jacob had the brainwave that if the colored material in squid ink was melanin, why not the colored material in other kinds of fossil material, fossil examples? So we went after an appropriate feather. Now, you should understand that the original interpretation of the kinds of structures that you'll see in a moment on feathers was that they were bacteria covering the surface. So well-preserved fossil feathers are covered in a series of little sausage-shaped bodies all over the surface of the specimen, which back in the 1980s were interpreted by Michael Wuttke, who's a German paleontologist, as bacteria feeding on the keratin in the feather. So if these things were bacteria, and we got a black and white feather like this one from the Crato Formation, we'd expect the bacteria to be all over the surface of the specimen. Whereas if they were melanosomes, these organelles that contain the color material, you'd expect them just in the dark stripes. And of course, that's given away the answer. Here's what melanosomes look like, both in the fossil and in the recent, and predictably, they're present in the dark parts and they're absent in the light parts. So Jakob got extremely excited about this, more excited than I'd have preferred, actually, in hindsight, but anyway. And went after all sorts of other things, but I took a slightly more step-by-step and persuaded him that there were things we needed to do first. So apart from looking at this, which was our proof of concept, this striped feather in the Cretaceous, let's leave this out. What we should then do is go to Messel, which is where Michael Wuttke had originally described these sausage-shaped bodies, and see if we could really prove that they were melanosomes and not bacteria, as Michael had originally said. Now, Messel, some of you will know, is an Eocene locality, not very far from the German city of Frankfurt, originally mined as an oil shale, and then proposed as a major dumping site for the region of Hesse, and a big hue and cry set up, and this is now being preserved as a fossil locality. So here's one of the famous mammals, and there are others. This is Microcranion, and you can see the outline of the fur, and this is what the fur looks like, little sausage-shaped bodies, which Wuttke would have said were bacteria, and we would now, I think, say were melanosomes. So this is the pit. As you can see, it's beautiful. It's essentially used for excavations, and it's a great big hole in the ground that would have been full of trash by now if the Germans had had their way originally, or some of the Germans. You collect this with the aid of student parties. They still do it every summer. You can saw it out of the pit with chainsaws. Literally, it's a waxy organic clay. Hump it around in wheelbarrows and then split it under an awning to stop yourself frying in the sun. A frog, again with the outline. A bat. There are spectacular bats at Messel. Mostly the fossils are transferred to resin in order to prevent them from cracking and drying. If you leave Messel shale on the surface of a bench or out in the open air, it curls up like an old sandwich very quickly and dries out and breaks up. There are birds, of course, many water birds, and isolated feathers. We didn't want to look at a feather in the resin, so we went after feathers that were still in the original shale. And to cut a long story short, the reason we know that these structures here on the surface of this Messel feather, and the slide is better than the light gives it credit for, we know these are melanosomes because not only are they color structures, in this case they're forming a thin film, which you can see it's about 2 melanosomes or 3 melanosomes thick on the surface of a barbule here, one of the portions of a feather, and that film is almost certainly evidence of iridescence in this particular bird feather, because iridescence is produced by light reflecting off layers inside the feather structure. So once you've done the Crato feather, where it was striped and you had a proof of concept, and you went to Messel and looked at whether or not these could be bacteria and demonstrated that clearly here was evidence of iridescence because you have a physical layer on the surface. The next thing to go after, of course, was dinosaurs, because we have this fabulous locality in the Jurassic Cretaceous. It's not really a locality, it's a kind of region northeast of Beijing in China, the Liaoning area, where they're pulling huge numbers of these spectacular feathered dinosaurs and indeed birds out of the Mesozoic. So this is the old Deinonychus. It's a Yale fossil, potentially, and here's how we would reconstruct Deinonychus now. So this is Bob Backer's reconstruction. Very live, agile, ready to rip your entrails out kind of reconstruction. And this is ripping the entrails out, but in this case of another dinosaur, a Tenontosaurus. But you can see that these little Deinonychus now have acquired feathers for insulation or whatever. I'm not going to go into the function. So you get these extraordinary reconstructions in the books. This is a book called Feathered Dinosaurs, The Origin of Birds by John Long. And this is based on a comparison between Avimimus, which is, of course, a dinosaur for which there is no feather evidence, and some kind of modern pheasant. And it's just showing you the potential that feathers can produce in terms of display. So the melanosomes have the potential to tell a story. And we were working on this in the States, and Paddy Orr and Mike Benton, Paddy here in UCD and Mike Benton in Bristol, with Chinese colleagues were working on this side of the Atlantic. And both of us managed to show that these Chinese dinosaurs preserve, where the feathers and other kinds of soft tissues are there, they preserve indications of melanosomes. So this is one of the illustrations from the paper by Paddy and Mike Benton and the Bristol group. And it shows some of the small structures that are interpreted as melanosomes in this particular dinosaur. What we were able to do was to get hold of a specimen of a late Jurassic form called Anchiornis, and actually take about 30 feather samples all the way around the specimen and put them under the SEM. And this was a preliminary effort. I'm sure this method will get refined as time goes on. But suffice to say that the packing and the size and the shape and density of these melanosomes differs on different parts of the dinosaur. And then you can compare this, details not important, this is a discriminant analysis done by Matt Shawkey, who's in Akron in Ohio, based on living bird feathers and the distribution of melanosomes in living feathers. And of course, essentially at a basic level, the long sausage ones tend to be dark black, the round ones tend to be russet or red, and then the combinations tend to give grays, or indeed where there are no melanosomes, you have the potential to identify white. So at a very simplistic level, you can plot your dinosaur distributions into this kind of diagram and produce a fluffy chicken. So this is Anchiornis, this late Jurassic feathered dinosaur, arguably the first, if you like, realistic, scientific-based reconstruction of color, at least in some kind of feathered creature. And I'm sure this will go much further. I've no doubt that both the Bristol group and I suspect people in China will work up this kind of method and look at other dinosaurs. And you have the potential to produce the sort of field guide to Jurassic and Cretaceous feathered dinosaurs in much the same way as you would use a field guide to identify the birds in College Park. So that's the feather story. So the final piece of this is what else do you do with exceptionally preserved fossils? You show the evolutionary history of soft-bodied groups. I started out by emphasizing that, you know, some 60, 70%, depending on how you count, of modern groups would not have any fossil record but for these exceptional preservations because they're entirely soft-bodied. So it's important to use soft tissues. So back to the paper. I'd have been stumped on this one because, of course, back in the 1970s, people weren't really thinking much about soft-bodied animals. I mean, the Burgess share was just coming on stream in terms of ideas and papers. I went off and worked on it in Cambridge. But since then, there's been a huge burgeoning of interest over the last 30 or more years in looking at these exceptional preservations. And part of that is because biologists have again become very interested with the coming online of genetic sequences and the interest in tree of life and biodiversity and the relationships of major organisms. Biologists have become very interested in the Cambrian explosion and in how these various metazoan groups set out on their route to the modern. So the fact that we have no question here we can identify that fits in with this evolutionary history of soft-bodied groups is in some ways a reflection of how the science has, or one direction the science has moved since then. So I'm going to go to Morocco just as my final illustration. As I said earlier, our Macaridian fossil came from Morocco, but more recently we've been getting ever more spectacular fossils out of there. And this is a group project. As I say, it involves Peter Van Roy, who did work here in UCD with Paddy Orr, Paddy and myself, and a number of other people around the world. And this material comes from an area north of Zagora. Zagora is an oasis town south of the Atlas Mountains in Morocco. So you fly into Casablanca, you get yourself to Marrakesh, you drive across the mountains and you end up in Zagora, and then you hire a four-wheel drive vehicle. And when you go and do this with Peter, he's always been doing it on the cheap. So you end up, I reckon, spending almost as much time getting the vehicle fixed as you do in the field doing field work, because you're driving across this rocky desert, which is, you know, really does beat up even these very hefty field vehicles. So this is Peter Van Roy and my student Jacob Winter, and this is myself. They gave me one of these blue headdresses, but I could never figure out how to tie the damn thing. It's pretty much the same as trying to do bow ties. This was almost as big a struggle as getting the lecture together. So this is what the field locality looks like. You've got to compare this working in the ore division of Morocco with working in the ore division of Wales or Wexford. The big difference is you don't have grass and fields and so on. You have virtually, well, not quite total exposure, but really very splendid exposure. And the story of this Fezuata formation, which Peter has discovered in the lower ore division of Morocco, is again soft-bodied. These are Burgess Shale type organisms. They're very much like the kinds of things that would be very much at home in Cambrian localities like the Burgess Shale. So that's one thing that's going on. They're persisting much later than we originally thought. But the other interesting thing about Morocco is that when you get this kind of extensive exposure, you pick up this exceptional preservation in many, many places. It seems to be virtually ubiquitous through a very substantial thickness of rock and over a significant area of 20 to 30 kilometers. So it's the same kind of story in a different context as Beecher's bed, where again, soft tissue preservation is much more common than you think. Here we are standing on a productive layer. This is Peter and Jakob, and I'm safely in the distance. So these are the kinds of fossils that we've pulled out of this. This is an animal called Furca, which is a little invertebrate, very much like Morella from the Burgess Shale. You can see that these things are pretty rare in later rocks, but a very nice example of something that's extraordinarily similar to an animal that occurs in the Cambrian. Here's one of Peter's arthropods, a thing he assigns to a colonielon type animal, and here's a horseshoe crab. So the other thing that's going on here is that not only are there Cambrian animals extending 30 million years later than we originally thought, but there are also much more modern creatures that these discoveries are pulling way back in the rock record to a much earlier time. So these are essentially the earliest horseshoe crabs, and what Peter has been able to show is not only are they the earliest horseshoe crabs, they're also extremely modern in aspect because they have a fused rear part just like a modern horseshoe crab. Of course, when I talk about horseshoe crabs in the United States, everybody's picked one up on the coastline. That's not quite the same here, I appreciate. So these are not perhaps so familiar. And the other wonderful thing about these particular exceptional preservations is they're not just, of course, about soft parts. This is, in contrast to Beecher's bed, a very normal marine biota from the ore division. So it has the whole range of taxa, including some very spectacular echinoderms like this articulated starfish. And of course, these kinds of complete shelly fossils are also extremely rare. And we have some spectacular early echinoderms. And as a closing slide, I might say that there's no better way to work on these exceptional preservations wherever you are in the world, be it in Morocco, where it's impossible to do without the local collectors, or in New York State, where you can get accountants who love to go out in the afternoons or the weekends and find new localities in a way that we just don't have time to do. So this is Ben Said, who's a collector that has worked extensively with Peter Van Roy, who holds material from localities for us to inspect when we go out, or he goes out every year, and who retains the locality information. And Peter's built up such a good relationship with him that he takes him to the various localities so that we can locate this in terms of stratigraphy and geography, as well as having these spectacular animals. So thank you, podcast, and thank you, everybody else.